Hey, ladies and gentlemen, today is Wednesday, the 14th, and I'm here to talk to you guys a little bit about chapter 10.1 and chapter 10.2. It has to do with transcription and translation. So I want to start with this. Last class, we began looking at transcription, which is a necessary function of taking DNA, which is inside the nucleus, and copying a message from the DNA and getting that message out to the ribosome, where that message can be converted into a protein. So this picture is showing the process of transcription. You're going to notice a couple of things here. The process of transcription, first of all, is broken down into three parts: initiation, elongation, and termination. What you'll notice about initiation is that there are three pieces of building this RNA molecule. We have a promoter here, which is a segment of the DNA that indicates this is where the beginning of transcription should occur. Then we have the RNA polymerase building, adding nucleotides to the growing mRNA strand. Transcription is happening this way. And then we have a terminator sequence here on our DNA that indicates for the RNA polymerase to stop. So termination means to stop. And remember, RNA polymerase, it means to make RNA, right? To make a polymer is adding nucleotides or monomers to a strand, and then we're making RNA. So remember, initiation is, it matters which way we're thinking about 5' versus 3' ends. We talked about that last class. We learned that RNA is built from by adding to the 3' end. So we're adding to the 3' end here. And then the way that DNA is copied is it is copied from the 5' end to the 3' end. If you're not familiar with what the prime represents, that would be something to look back from last class, or just go ahead and Google it. What does 5' and 3' mean for DNA? And then finally, termination. Once that RNA polymerase gets to the termination site, the RNA polymerase will leave that site and allow that RNA to break free from the strand of DNA, allow that DNA to recoil, and that mRNA will now leave the cell. I want to point out that we are able to simultaneously transcribe DNA at several different points, that we don't have to have DNA unzip only for one gene at a time. DNA can have several sites of transcription happening all at once, and it's called simultaneous transcription. Remember too that RNA is short-lived, meaning that once this mRNA is transcribed, it does not last very long within the cell. And so it has the immediate job of leaving the nucleus site and heading out to the ribosome to start building proteins. When we're thinking about RNA processing, we want to remember that mRNA has anywhere between 5,000 to 4,500 nucleotide bases. Now, I want to remind you, if you look at this picture here, only a piece of that is actually the protein coding sequence of the gene. We have other pieces of that mRNA strand that have basically jobs that tell the ribosome what to do. So we have what's called a 5' methylated cap here at the end. It's essentially kind of like an indicator for the ribosome to start or stop. And then over here, we have a poly A tail, which is essentially between 50 and 250 adenine nucleotides that are added to the 3' end. And again, these two ends help to kind of direct the cell what to do once this gets to the ribosome. That methylated end and that poly A cap help to direct the ribosome what to do. There we go. Now, what we had today to look at a little bit more closely is this idea that even after we have a protein coding sequence, we can splice out sections of that protein coding sequence to change slightly the function of the protein. What that means is within that protein coding sequence, we're going to have kind of two things we're looking at: introns and exons. Introns are segments of those coding sequences that need to be removed. Introns don't serve a function with the protein building. And so what scientists believe is that those introns perhaps are remnants of ancestral genes that are no longer needed. And so before we can build a protein, we need to get in there and get rid of those introns, because they don't make sense with the proteins that are needed for that organism. So introns are removed and exons are the parts of mRNA that remain. And those are the pieces that are spliced back together and translated into amino acid sequences. And you can see that here. If you look at the picture, we've got the arrow here is pointing at the exons, those three kind of darker pieces. And then the introns there, we splice out. They basically get resorbed by the cell and are no longer needed. There's a function called alternative splicing, and this is where once we've got those exons and introns kind of sorted, so we've got those exons that are now going to be used for the protein coding sequence. Well, what can happen within that is there can be something called alternative splicing, where only segments of those exons are used. So only certain exons are used for certain proteins, and this will have a slight effect on the protein function. And I think the example in the book had to do with liver proteins versus another protein in a different organ that basically has to do with absorption. And so if we have a protein whose job is absorption, it's still got the main function, but there might be a slight difference in the protein assembly to make those proteins for absorption just very specialized for that organ. So protein A might be an absorption protein for, let's say, the liver, whereas protein B might be an absorption protein, say, for the small intestine. And so they're still absorption proteins, but with slightly different functions, and so slightly different arrangements of those amino acids or sequences. Now, this leads us to section 10.2, which is about translation. So once we have the gene that we want, once we've spliced it in a way that makes sense for the function that we're going to be using it for, we now have the ability to take that sequence and turn it into a protein. So this is what translation is. It's where we have that sequence, it's ready, it's methylated, it's got the start and stop signals there, and we're ready to turn it into a protein. So particular mRNA codons are going to correspond to particular amino acids. What that means is within those exons that we have spliced out and now arranged in the way that we need, what happens is within those exons, there's going to be sequences of 3 bases, so 3 bases at a time, and those 3 bases are going to correspond to amino acids, which are the building blocks of proteins. So if we're looking at the overview of translation here, we're looking at, here's our DNA template strand where we were inside the nucleus. This is transcription happening where we have the messenger RNA being created inside the nucleus. Now that messenger RNA, which is green, is now going to be broken into 3 nucleotide chunks. So each chunk of 3 nucleotides is called a codon. So those codons, for example, AAG, the letters AAG correspond to an amino acid, and that particular one is lysine. And remember, proteins are basically a long chain of amino acids, and so each 3 nucleotides, which are called a codon, relate to different amino acids. I believe there's 64 of them, and the arrangements of amino acids in the protein chain are what determine what the protein function and structure will be. The way that this was figured out is actually pretty interesting. So when scientists in the 1960s were looking to figure out how DNA is translated, they didn't know that bases of 3 were used to match to corresponding amino acids. And so what researchers did was they used logic and little tiny clever experiments on simple genetic system to decipher a code. They ended up using viruses because their genetics are rather simple. And they added and removed bases from a viral gene, just basically one at a time, 1 base at a time, then 2 bases at a time, then 3 bases at a time. And what those researchers found out was that if they removed 3 bases from that viral gene, it had less of an effect on the protein overall. And so they were able to deduce that by removing 3 amino acids and specifically 3 nucleotides and specifically 3 nucleotides in a certain reading frame, they figured out that removal actually had less of an effect than removing 1 or 2 bases. And it took a long time to figure out, but eventually they figured out that there's something called a reading frame when it has to do with translating, and that's what we're going to look at now. So a reading frame is basically this idea that the mRNA strand with its many bases has to be read at a certain within a certain window. And what that means is we have to make sure that we start reading that RNA in the correct spot. And that's through the use of something called a start codon, which we'll talk about in just a moment. Essentially, the ribosome looks for a start codon. And after that start codon, so it's, what is it? AUG is the start codon. So after finding the AUG start codon, essentially the ribosome sets up a reading frame. So after AUG, it's going to read the next one, ACG. Okay, then the next one, and I'm looking right here on this graphic here, and then GAU. And so every 3 nucleotides after that start codon represents basically those 3 nucleotides within the reading frame that are translated into those particular amino acids. And you'll notice the reading frame also includes a stop codon, which is also very important so that the ribosome knows when to stop adding amino acids to that protein. Here's a table that shows all the amino acids that are available. Now, something really interesting is even though organisms on Earth are highly diverse, all organisms have the same genetic code. And what that means is if we have a UUU, for example, in any organism on Earth, UUU as a codon is going to relate to phenolphthalein, which is an amino acid for all organisms. So if we look at this chart, cysteine, UGU, cysteine is going to be UGU in humans and cats and frogs and snakes and spiders and plants and dahlias and everything. So this is we call it a universal genetic code, meaning all organisms use these sequences as the sequences for amino acids. We say that the genetic code is a triplet code. We say that the word triplet just means 3. So a codon is represented by 3 bases. There's 64 total codons that we use to represent amino acids. And altering the DNA sequence by even 1 or 2 bases produces a different amino acid. So if we disrupt the reading frame, as in if we don't read from the start codon and we just jump right in, this affects every subsequent amino acid. And this is going to be something we talk about when we look at mutations. All right, so I got my red slide here. That means I'm going to be stopping for today. What you're going to do, hopefully, is take an opportunity to supplement your notes from chapter 10.1 and chapter 10.2, and hopefully be leaving me some discussion questions that we can talk about in class. I hope you guys have a great day. I will talk to you soon.